Could it be that it is time for another biology department video? Yes, it is. It is. So good news all round. And this one is on the topic of transport in humans. How we transport things all around our body. Now, of course, you know by heart, by this stage, the equation for respiration, don't you? Yeah, yes you do. And the equation for respiration is oxygen plus glucose goes to carbon dioxide plus water. Now, of course, this is going to go on inside every living cell in your body. It's going to go on inside specialized organelles called the mitochondria. And in order for them to do this, they're going to have to receive glucose and oxygen from somewhere. And how's it going to get there? Well, it's going to get there from the lungs, if it's oxygen, via the blood. And if it's glucose, it's going to get there from the intestines, via the blood. And that's what we're talking about now. And of course, we mustn't forget that we've got to get rid of all that carbon dioxide from respiration as well. And that's got to get to the lungs via the blood. So let's start off by thinking about the blood. And really, we're going to split up this topic into two videos. The first of which will largely be thinking about what the blood does, and the second of which we'll be thinking about what the heart does and what the blood vessels do. So, let's press on. Our blood. Well, an average-sized person, I suppose by average size we mean adult, male, normal sort of size, probably about Mr. Dare size, that's what we would guess, has about 5 litres of blood. And that is a very special type of tissue consisting of millions of cells, and it does these two main things. It has its big role in transport, and that is transporting oxygen, as we've said, nutrients, glucose for example, carbon dioxide and urea, those are both waste products that need to be transported off for excretion, hormones, and heat energy. Heat energy we wouldn't count as a substance, of course, but it does have to be carried around. If your muscles are working hard because you're running and you generate a lot of heat energy in those muscles, we need to move that heat energy before it damages those muscles, and the blood will do that for us. Its second role is to help to defend the body against disease. The blood's made out of two main components, the plasma and the blood cells. The plasma just being essentially the watery solution that suspends the blood cells. And the different parts of the blood have this function. Here we go. Components of blood. Plasma. Description of components. It is a straw-coloured liquid, mainly made of water. You may have seen this before. If you've had a cut, and it's, it's a small cut, and it's healed over, and you've had a bit of a scab on it, and maybe the scab slightly oozes a little bit of fluid, that little bit of fluid is probably a kind of yellowy straw colour. Well, that is plasma. That is blood with all the red blood cells filtered out of it. So now you know what plasma looks like. And its function is, as we just discussed, to carry mineral ions, glucose, hormones, amino acids, carbon dioxide, and urea, and carry heat energy around. Red blood cells, they have a biconcave shape, and we'll look at that shape quite soon. They have no nucleus, which is a little bit strange for an animal cell. Actually, they are made with a nucleus, just as these red blood cells, which are made in the bone marrow, mature. They lose the nucleus that they were made with when they started off in life. And they contain hemoglobin, and their function is to contain that hemoglobin, which itself will carry oxygen from the lungs to the organs. Our white blood cells. Now, white blood cells are a bit bigger than red blood cells, indeed considerably larger sometimes than red blood cells. And they come into two categories, the lymphocytes and the phagocytes. Lymphocytes, large round nucleus, and phagocytes are large as well, but they can change their shape because they can need to squeeze through gaps sometimes, and they have a lobed nucleus. We'll get onto that again. Lymphocytes, they produce antibodies. They are the magic bullets, those antibodies, the magic bullets of our immune system. They will seek out invaders and shoot them down, whereas phagocytes will engulf foreign bodies like the blob. They will slither over them and engulf them and digest them and do horrible things like that. Platelets, they are fragments of cells, not real cells, fragments of cells. They don't have a nucleus, they are only a fragment of a cell, and they are pretty titchy, and their role is to clot the blood. So, red blood cells, well, made in the red bone marrow, they live about 100 days. And then they're destroyed, broken down by the liver or spleen, and their hemoglobin is broken down into a couple of pigments, bilirubin and biliverdin. Not words that you need to know, but bilirubin is the substance which gives our blood that straw colour, and incidentally gives your urine that straw colour as well. And they only have one function: to transport oxygen. Okay, not strictly true, but let's just think of it as true. They only have one function: to transport oxygen. Not that they don't transport oxygen, they certainly do. They've got some side functions as well. But yes, just one function: to transport oxygen. And this is what they look like. Well, they look like a kind of rounded off refresher. You will remember refresher sweets, I'm sure, if they're still called refreshers. Just like a rounded off refresher. They are a biconcave disc. Now just look here. I'm going to see if I can change my pen to another form. Here we go. So this is the biconcave shape that we're talking about here. And what it means is that we have a larger surface area of this cell for its volume. So it gives it a really big surface area to volume ratio. Let's have a look at its features. It has no nucleus, and that means you've got more room for hemoglobin, so you get more oxygen carried. Lots of room. So, no nucleus, more room for hemoglobin. It contains, hey, hemoglobin. I think we just said that, didn't we? And we've got that hemoglobin, and its role is to bind easily to oxygen. It does that in the lungs, picks it up, and then it can carry it off and dump that oxygen at the tissues. And it has this biconcave disc shape. Well, what does that do? It gives it a higher surface area to volume ratio. What on earth does that mean? Well, it's got a large surface area for its actual volume, and that just means that oxygen diffusion happens faster, which is good news. So their function, well, particularly the transport of gases around the body, particularly oxygen, which I've just circled very poorly there, but never mind. And oxygen concentration is obviously really high in the lungs, and so in the lungs, hemoglobin, the red blood cells, combine with oxygen to form this substance, oxyhemoglobin. So they pick up oxygen in the lungs, and then they carry that oxyhemoglobin from the lungs to the tissues. Now you'll remember from Nature and Variety of Living Organisms that tissues are collections of similar cells doing a similar job. So they carry oxyhemoglobin to your cells, and those cells are respiring, and they need oxygen, and then they let go of the oxygen. The oxyhemoglobin lets go of its oxygen, and that oxygen enters the tissues by diffusion, becoming, again, oxyhemoglobin. Let's have a look at that. Well, we've got the lungs taking up the oxygen, we've got oxyhemoglobin here, and then as that oxyhemoglobin travels to the respiring tissues, it drops its oxygen, it dumps its oxygen itself. It then becomes hemoglobin, and it goes back to the lungs to pick up more oxygen. The blood also carries carbon dioxide. Now, carbon dioxide is the product, or one of the two products, of aerobic respiration. And it's got to get out of the tissues. It's acidic, and if you leave it in the tissues, it will really damage those cells. And so we've got to carry that carbon dioxide, that CO2, away from your tissues and take it off to the lungs. And it's the lungs which are going to get rid of that CO2. And here's our equation for respiration. Here we go. We're going to do the full chemical formula for respiration. Now I'm going to see if I can write it with this pen. Here we go. C6H12O6. Now that is the formula for glucose. Plus 6O2 goes to 6H2O, that's water, plus 6CO2. That's carbon dioxide. That's our equation. So we've got to bring glucose to the cells. That's done by the plasma. And we've got to bring oxygen to the cells. Done by the red blood cells. Then we've got to get rid of, well, the water. Well, there's plenty of water sloshing about anyway, everywhere, to be honest. But we've got to get rid of this CO2. We've got to get rid of that. And it's carried dissolved in the plasma till it gets to the lungs, and then it diffuses out of the alveoli. And more about alveoli in another video, I'm sure. That was the red blood cells. Right, white blood cells. Larger and fewer of them. 8,000 per millimeter cubed of blood. That's still quite a lot, isn't it? 1,000 millimeter cubed of blood. Yeah, quite a lot. And their function is to defend the body against disease. Right, so what do all these things mean? A disease. What does a disease mean? Well, it's a condition that prevents the body or part of it working properly. And we can recognize different diseases because of the symptoms they give us. And symptoms is just another word for signs. So if you're being a bit sick and doing unmentionable things, then that is a sign that you are unwell, and it'll be a sign of a number of different things that could be causing this particular condition or disease. And most diseases we get are caused by other organisms, although that's not exclusively true. Sometimes it's our body itself which just decides to go wrong for various reasons. But most diseases are caused by other organisms. They get into the body and they feed and reproduce inside the body. Now remember that plasma. What's in that plasma? The plasma is a watery solution with loads of glucose, lots of oxygen sloshing about, loads of amino acids. It is about the most perfect place that bacteria could reproduce. If you were trying to grow bacteria in a lab, you'd think, I need some water, I need some glucose, I need some amino acids, maybe throw a few vitamins in there, and keep it nice and warm at 37 degrees. And of course, that is our blood. So our bodies are just wonderful places for bacteria to live. And as a consequence, we end up getting colonized by these things which excrete toxic waste substances and damage our body and just release all these horrible toxins. So we've got to fight off against them, and that's what our white blood cells will do. Viruses, bacteria, particularly potent, and how do we fight against it? Well, first of all, we've got our skin and our membranes of the body, and they will act as physical barriers. So our lungs are lined with alveolar epithelium, and that will act as a barrier against bacteria and viruses. Likewise, our skin is a really effective barrier. We've got other barriers in various other places, such as your eyeballs. Your eyeballs have got a barrier, and they secrete lysosome, which will act as an antibacterial agent. So everywhere you've got a contact between you and the outside world, you've got to have a physical barrier stopping bacteria from getting in. That's what your stomach acid is about. That's what all that acid is in your stomach. It's not particularly to help in digestion, it's to kill bacteria in your food. But then, let's say you get a cut. Then white blood cells can come in and they can take over the defense. So they go for it. So two main types of white blood cells. They are the phagocytes and the lymphocytes. And let's have a look at them. Phagocytes, right. Made in the bone marrow, red bone marrow, live only for a few days. And their structure, well, how do we describe them? They are irregular in shape, with an unusual shaped nucleus and cytoplasm containing granules. Let's have a look at that. Here we go. Phagocyte diagram for your notes. Look at this. It's not the best one, is it? I'll grant you that. Yeah, it's a kind of a bit scaled up from a smaller image. Just really cell membrane around the outside. Here we go, and it could be any shape really. And then we've got this kind of funny shaped nucleus like that. That's our funny shaped nucleus. And what have we labelled it? Well, we've got our granular cytoplasm there. We've got a lobed nucleus, and if you've been looking at these under a slide, you'll remember these lobed nuclei. And of course the cell membrane, which is just this thing here. That's the cell membrane. There we go. I'll write it there. There we go. That's where you can see it. And what do? Well, they destroy foreign substances such as bacteria by engulfing or eating them. Let's have a look at some processes. Excuse the quality of these images here. You'll just have to live with it. Live with it, will you? Stop complaining, please. Right. So let's say we've got these bacteria here. It says bacterium there, but very clearly there are a few of them. There are 7 of them there. And so, plural of bacterium, bacteria. So I'm just going to write an A there. No, I don't want you. There we go. I'm just going to write an A there. There we go. And then this phagocyte will start doing clever things with its cell membrane. And it will reach out with these, well, they're called pseudopodia. Pseudopodia means false feet, and amoebae have them as well. You don't have to remember the word pseudopodia, but it's quite a cool word, I think. And they reach around the bacteria with these pseudopodia, and then they engulf them in this vacuole here. Now, you will remember from your cells course that animal cells don't have vacuoles. Well, it's obviously not true. It is certainly true that animal cells don't have the large vacuole that plant cells have, but animal cells do often have these mini vacuoles like this. In fact, it's drawn much larger than actually it would be in a real cell. But we do have vacuoles which contain fluid, and here it's engulfed these bacteria and engulfed it in, well, a vacuole. And then what it does, it chucks a whole load of horrible digestive things into it, digestive juices, as well as some other stuff, some other particularly nasty stuff like hydrogen peroxide, H2O2. You'll look at the formula H2O2 and think, I know H2O, that's water, but there's an extra oxygen on there. Yes, and that extra oxygen is really, really damaging. And so that's just one of the nasty things that we use to destroy bacteria with our digestive enzymes, which makes it all good and keeps us safe. Let's have a look at how that happens. Here, this is cool. I really like this. This is a phagocyte here. So we're going to write hero on the phagocyte, and we'll put exclamation marks on both sides of that. And that is engulfing a yeast cell. Yeah, look at that. Yeah, I haven't seen this before. I've seen this image before. I haven't noticed this before. That is a scar where the yeast cell has done budding, where it's done a little bit of mitosis, a bit of cell division. Anyway, that's beside the point. Our hero, our phagocyte hero, is engulfing this yeast cell here. And over here on this image, this is just a favourite of mine. The bacteria have been labelled with an antibody with a fluorescent tag on it. So we can see where the bacteria are, and we can see also the phagocyte cell membrane has been labelled with a different coloured fluorescent tag, so we can see them. And you can see bacteria just being nabbed by the phagocyte, and there you can see it's actually inside one of these vacuoles. So, and then here we take in more. The time frame goes dum 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 dum, oh dum dum, like that. So it just keeps on wolfing down these bacteria until eventually it is just jam-packed full of these bacteria. And what it will do, it will secrete digestive enzymes into them, break them down, digest them, kill them. That'll be job done. And well done. We applaud that phagocyte for doing that. And this, I just think, is absolutely astonishing, this picture here. These are bacteria. And this is our phagocyte, and we'll write hero again. Here we go. And it is lassoing these bacteria. It is sending out a lasso. I can't describe it in any other way. A lasso to pick up a whole clump of bacteria and drag it in. And this is not a computer-generated image. This is a scanning electron micrograph. This actually is what happens. You are looking at a photograph of what happens. You can pause the video at this point and just go, wow, isn't that amazing? And then restart the video. I would recommend you do that. Okay, you've done that. Yes, you've done it. Good. So phagocytes, what else can they do? Well, they can change their shape, move from place to place. Therefore, they can squeeze through the walls of the capillaries into the tissues. You'll learn about capillaries in the next video. And that means they can move. They can move to areas of infection, such as a cut, and attack the bacteria at this site. Okay, minor gore coming up. It's a photograph of my knee after a football match. It's gone. It's gone. Oh, that's horrible. Okay, yeah, you can see that's minor gore, and you can clearly see that is a nice entry point for bacteria. So you need your white blood cells to deal with that. Of course, you also need platelets to clot it afterwards. More on that later. Okay, the gore's gone away. You can relax. Right, lymphocytes, next one. These are made in the lymphatic system, and they can live for several years. Now, different from phagocytes, which are short-lived. And they're spherical in shape, very large nucleus, and a small amount of cytoplasm. Here we go. Again, you may remember seeing these when you looked at them on your slides, if you did so, on a microscope. And it's got this huge nucleus here, hasn't it? This large nucleus in the middle, and only an incy-wincy bit of cytoplasm. Here, of course, these are red blood cells, and you can see, this is how you can see that they're biconcave. They look a bit lighter coloured in the middle, and that's just because they're thinner in the middle, and so the light shines through them more easily in a light microscope. Oh, here come the labels. Draw a diagram of this lymphocyte into your notes. There we go. Small amount of non-granular cytoplasm and large nucleus. Hooray. Now, our lymphocyte does not engulf foreign bodies, but what it does is it makes these magic bullets, these antibodies, and these antitoxins are magic bullets. What do they do? Well, these antibodies, the antibodies that is, destroy foreign substances, such as bacteria and viruses, and they've got these effects. They can make the bacteria and viruses clump together, and then they can be engulfed by phagocytes. Let's just flick back a couple of pages. So, here we go, flicking back to this. Now you see these bacteria here, they're all stuck together, and they'll be stuck together because antibodies have made them do so. And that makes it easier for our phagocyte here to lasso them and drag them in. So you can see, if you get all your bacteria clumping together, that's useful. Okay, here we go. Careful of the minor gore coming up. Oh, there it is. Okay, gone. And back. They can themselves make bacteria and viruses burst. And yes, as described, the clumping together makes it easier for phagocytes to engulf them. Separate point. Since certain bacteria and viruses do their damage by secreting toxins, what some lymphocytes are able to do is they can secrete special antibodies called antitoxins, which will neutralize these toxins. They neutralize the toxins. That's good. That's good for us. Almost there in this video, so let's press on. Vaccination and immunity. Now, when we encounter a pathogen, remember the definition of pathogen. A pathogen is a microorganism which causes harm to us. It causes harm. And when we encounter one, our lymphocytes generate antibodies against it. And to prevent the body being infected by the same pathogen time and time again, lymphocytes also produce memory cells. Key word here, memory cells. And these cells stay there. They remain in our circulatory system for a lifetime. So what? Well, the next time our body encounters that same pathogen, the memory cells react to that pathogen very quickly. Now, those memory cells are still there. That's why it's so important they last a long time. They're there. They react to this pathogen that they've met before, and they make antibodies much earlier than the first time. Much earlier. So it's just faster. Much faster. And that prevents us being infected twice by the same pathogen. We are said to have gained immunity. Let's think about vaccination. And it works on this principle. First of all, the body is given a vaccine. What is a vaccine? Well, it's a dose of the pathogen. You just give someone a bit of pathogen, normally by injection, but they're developing nasal sprays now for the flu vaccine, so not exclusively by injection. And that dose is either number one in the red corner. Well, they're both kind of in the red corner, aren't they? Number one, the dead pathogen. You kill the pathogen and you inject it into someone, hoping that it doesn't come alive again or do something awkward like that. And then you can see it. You can see that dead pathogen. Or you might chop the pathogen up and inject people with parts of the pathogen. Why do we do that? Well, the lymphocytes can generate antibodies against the surface of the pathogens. On the surface of every cell, there are substances called antigens. We can't help but have antigens on our surface. If our white blood cells meet one of our cells, some of the antigens will say, "Ho, ho, ho, hi there, you know me, I'm part of you," and they'll be recognized as self. But if you've got a pathogen in there, it will have antigens on its cell surface which do not say that at all, which say, I am foreign, I do not belong in this place, and our lymphocytes will recognize them and they will target them. And so if we can just inject someone with the antigens without the pathogen itself, or maybe if the pathogen is dead, so we just inject the pathogen cell surface, well, our lymphocytes can make antibodies against that pathogen, against the antigens on that pathogen. And that means they can make memory cells. And what do we say about those memory cells? Well, they would last in the body for a long time. And that way, the next time your body meets the antigens from this pathogen, it will recognize it because we've got those memory cells. And very quickly, we can make more antibodies against it. And that's the way that vaccines give us immunity. Very important point, the antibodies themselves don't hang around in your body for ages. What do hang around in your body for ages are the memory cells, which can then go and make the antibodies very quickly the next time you meet a pathogen. Final little point on this video are about the platelets, and these platelets are tiny fragments of cells. They're made in the red bone marrow, and they're approximately 400,000 per millimeter cubed of blood. And you can see just on this image here, their sort of size. They are, as I say, cell fragments. There was one there. And their role is simply to clot blood. This is a blood clot here, and these are platelets kicking out a whole load of protein strands. Essentially, they make proteins which are normally in your blood and are dissolved and go around your blood perfectly easily. They make those proteins bind together and coagulate. Now that forms a whole mesh around a cut, around the damaged area. And that mesh traps, you see this red blood cell, which is trapped there at the moment, and therefore that is going to plug the gap. So you can read this sheet and that will take you through. So pause there as you wish. So once you've read this sheet, that is the end of this video. I'm going to put a couple of tutorial questions up in just a moment, so get ready for the tutorial questions. Thank you. So here are some tutorial questions. Do try to answer these. What are the adaptations of a red blood cell to maximize its carriage of oxygen? How do phagocytes destroy invading pathogens? The lymphocytes produce antibodies. How do these molecules destroy invading pathogens? And this one will probably take a little bit more detail to explain. Explain how vaccination leads to immunity. Thank you.